Welcome back to another virtual food safety office hours. This is the last one of 2025, right before the holiday break. I am Abby Snyder. I'm an associate professor of microbial food safety here in the Department of Food Science at Cornell University. And I'm joined by my colleague, Assistant Research Professor Dr. Nicole Martin, who also directs the Cornell Dairy Food Center, and Dr. Kristin Schill, Assistant Research Professor at the Food Research Institute at UW-Madison. So today, our aim is to address some of the work that's being done on infant formula safety. In particular, we've had a couple notable recalls in the past few years related to formula. So one in 2022 for Cronobacter, and most recently, the ByHeart recall due to cases of infant botulism. Although the investigation into the ByHeart case is still ongoing, we're going to provide some updates on what we know so far, what is different between the current and previous recall based on differences in the pathogen, and differences in risk messaging for infant botulism compared to the Cronobacter cases from before. And so, what we're going to each do is give a brief presentation, and then we're going to save questions till the end. I do have all of the questions that you pre-submitted with registration, but I can also take questions if you want to direct message me through the chat, so that would still be confidential if you like, during the talks or during the Q&A portion. So with that, I'm actually going to kick it off by turning it over to Kristin.
Okay, thank you very much. And hopefully you can see my slides. So I'm going to go through a little bit of the update today. I'm going to move right along. Oh, if I can move my slides.
I am not seeing the slides. Is that just me?
Do you see them
I'm
now?
not seeing them either.
Hmm. All right, let me.
There you go.
Okay, so now you can see everything. And we tried this out right beforehand and everybody could see it. So, so it's funny. Okay. So, yeah, thank you for including me on this discussion today. My role is to go through, briefly go through the Cronobacter sakazakii recall in 2022, and then provide an overview of the botulinum toxin producing species of Clostridium and botulism, and what the disease is, and kind of give you a little bit more information of the organism, what organisms actually cause botulism through production of the toxin, of the botulinum neurotoxin, and then kind of go over what we know so far by the ByHeart formula that's implicated in this infant botulism cases. I'll also provide some background and some historical considerations for Clostridium botulinum and powdered infant formula. And then just a last slide on our capacity for seed botulinum work at UW-Madison. So our lab is actually one of the very few labs in the US that can work with Clostridium botulinum and its toxins. But I'll get into that later. So for the Cronobacter sakazakii outbreak, this was really detrimental happening coming out of the pandemic. I just want to provide a little statement, though, that everything that I'm presenting today is general knowledge. It is something that's already been publicly available. So I just wanted to put that out there. And I certainly don't want to be blasting any companies. This is simply what happened. So back in 2022, the CDC was notified of consumer complaints of some infant illnesses related to powdered infant formula products that were manufactured in Abbott Nutrition in Sturgis, Michigan. So in September 16th of 2021 through 2022, the CDC had received multiple reports of these 3 Cronobacter cases in infants, and later they identified another case for a total of 4 cases. And unfortunately, there were 2 deaths that also occurred, and this was in 3 states within the United States. There was an initial complaint of a salmonella infection, but that was later deemed to be not included in this product or in this situation linked to this outbreak in particular. So on February 17th of 2022, Abbott Nutrition initiated a voluntary recall of certain powder infant formula products. FDA had initiated on-site inspection. They did find positive Cronobacter samples from environmental samples. They found 5 actually distinct different strains, but product samples that were collected by FDA tested negative for Cronobacter. And CDC, obviously, as part of their outbreak investigations, they performed whole genome sequencing on the patient isolates. And they were not found to be genetically related to the strains that were found in the environmental samples from the company at the processing facility in Sturgis, Michigan. And also the patient isolates were not closely related to one another. So a couple things to point out, this is not, at the time, Chronobacter was not a notifiable pathogen, and there was not a lot of whole genome sequencing that was being conducted at the time. So we didn't have a lot of other samples to compare it to, like reference genomes to compare it to. So in March of 2022, FDA released the FDA Forms 483s from the 3 inspections that were conducted at that facility. You can go online and download those and read those for your information. But Abbott did not establish a system. This is what FDA was stating. They did not establish a system of process controls covering all stages of processing that was designed to ensure that infant formula does not become adulterated due to the presence of microorganisms in the formula or in the processing environment. Abbott Nutrition also did not ensure that all surfaces that contacted infant formula were maintained to protect infant formula from being contaminated by any source. So what Abbott had done, they conducted standard product testing, but with limited numbers of samples. And so finished product testing doesn't actually eliminate the risk of contamination, but it provides greater chances of detecting Sakazakii and Chronobacter. And so knowing once you detect it and you know that it's there, then you can better sanitize and prevent the organism from coming in contact to your food product. In 2024, Chronobacter did become nationally notifiable pathogen, so doctors and labs were then required to report cases to health departments because even though Chronobacter is so rare, it can lead to fatalities among infants who consume product that's been contaminated with Chronobacter. Before that, FDA had relied on consumer complaints of illnesses and that were found in the patients. And again, WGS or whole genome sequencing was rarely performed on those isolates. FDA did come out with information of how to safely prepare infant formula and how to store powdered infant formula safely to prevent further outbreaks. But this was really kind of a matter of sanitation and having that product being contaminated with Chronobacter. So I'm going to go into a little bit of an overview of Clostridium botulinum. This is different in the sense that spores are typically everywhere of Clostridium botulinum, but this is an organism that produces heat-stable endospores. Historically, anything that produced botulinum neurotoxin was considered Clostridium botulinum, but we know through a variety of genomic techniques that there are multiple different species and groups of organisms that can produce botulinum neurotoxins, and these are designated A through G. And there's also X and HA variants and DC variants and hybrids and things like that. The main thing, though, to take into consideration is that it's mainly group 1 and group 2 and group 5 and group 6 organisms that will cause botulism in humans by producing toxins. Group 3 has not been linked with causing disease in humans, but causes disease in animals. And argentinens, which is group 4, they haven't been linked to causing disease in humans as of yet. But the group 1 strains, these are the strains that have higher heat resistance, the spore heat resistance. And so spores are everywhere. They're geographically distributed. And so we always just assume that there's going to be a low level of spores that could be present in a food product. And so primarily control of Clostridium botulinum is through either killing all of the spores through retort thermal processing or other thermal inactivation strategies to inactivate spores of Clostridium botulinum, or by formulating the food product to prevent these spores from germinating, growing, and producing toxin in the food product. Where foodborne botulism is from the consumption of preformed toxins in the foods. And we do that through addition of preservatives to prevent the organism from germinating and growing and producing toxins, lowering the pH, lowering the water activity, lowering the combination of pH and water activity, things like that, in order to prevent the organism from growing and producing toxins. I also want to point out that Clostridium sporogenes, which is located in group 1, it's genetically very, very similar to group 1 proteolytic strains. And this is going to be important for a slide later on, which I'm going to show. There are strains of sporogenes. It was believed that sporogenes was mainly non-toxigenic, but there have been strains, 21 strains that have been found to produce toxin. So I just wanted to point that out. So what is the disease botulism? Because sometimes in news articles you see, oh, there's botulism in the powdered infant formula. Well, it's not botulism that's in the powdered infant formula. It's Clostridium botulinum spores that are present, and botulism is the disease that is caused by the toxin that's produced by Clostridium botulinum or botulinum neurotoxin producing species of Clostridium, because there's a bunch of different ones. So botulism is an acute poisoning caused by botulinum toxin. It's a descending paralysis, so it starts with the facial and ocular muscles. So there's blurred vision, difficulty speaking and swallowing, and then leading to respiratory failure eventually, potentially. And before the 1950s, there was a high mortality rate. Now we've been able to, through the administration of antitoxins, botulinal antitoxins, and supportive care, reduce that mortality rate down quite a bit, significantly. There are other types of botulism. So I've listed those here on the left-hand side. There's typically botulism in comparison to other foodborne outbreaks. Clostridium botulinum and botulism in general is fairly rare, with about 100 to 200 cases reported annually in the United States. Infant botulism, which is from the ingestion of spores of the organism, that accounts for the majority of cases, unfortunately. So around 65 to 75% of the cases are infant botulism cases. Foodborne botulism constitutes for only 15 to 25%, and this is ingestion of the preformed toxin in a food product. I put together a little figure that's on the side here that shows just infant botulism, foodborne, and wound, because wound botulism, these are the three most common types of botulism. Infant botulism over the last 10 years or the last 25 years has been increasing significantly. You'll see that around from 2002 through 2010, it was around 80 some odd cases, 80, 90, 100 cases. But then once we hit 2011, it went up to about 120, 130 cases, and then it just kind of skyrocketed from there up to about 181 cases in 2021, 2022. So what's important is, what is the cause of the increase in infant botulism cases? Is it due to just better detection? It used to be in the 80s, 70s, and 80s and 90s, sudden infant death syndrome. But then it was later thought that, well, some of these sudden infant death syndrome cases are actually botulism cases, infant botulism cases. That was before infant botulism was actually characterized as a disease, I think in 1978. So there is an increase now. Maybe it's better detection, or what are we doing differently with regard to processing food products, infant formulas or infant food products? We don't know. Maybe nothing. Maybe it's just a matter of better detection. Foodborne botulism has stayed the same over the years. There may be some spikes with larger outbreaks, but typically it has been staying the same. Wound botulism has increased due to increased use of injection drug users. Adult colonization botulism, I do want to touch on that. It's fairly rare, but it has been, it's very similar to infant botulism. And that's the ingestion of the spores. For adults, normal, healthy adults, we consume spores every day. If you eat carrots or onions or vegetables, raw vegetables, you're consuming spores, but it's not going to be detrimental to your health if you're a normal, healthy individual, because you have an established gut microbiome or a microbiota. So bot doesn't like to compete, so you're fine. But under certain situations where a patient has undergone extensive antibiotic treatment, where you have wiped out your gastrointestinal microbiome, then you will end up with the potential of having adult colonization botulism if you eat a particular food product that may have a high load of spores. So I just want to kind of clarify what the differences are. So infant botulism, again, is the ingestion of spores of Clostridium botulinum, which in the absence of a gut microbiome can germinate, grow, and produce toxin within the intestine. This usually affects babies who are between 2 weeks and 6 months. But all babies are at risk until their first birthday. And it's been over the years, they've identified honey as a risk factor for infant botulism because of the high load of spores in honey. That's why you don't feed honey to infants for less that are less than a year old. Other sources of spores have been linked to corn syrup, soil, and dust. I would say, though, a majority of these cases of infant botulism, or a large portion of infant botulism cases, the source ends up going unnoticed. And so soil or dust may maybe be attributed to that. But it's really hard to track down what the cause of infant botulism is in many, many cases, unfortunately. The fatality rate is about 2%, and this has been dramatically reduced. Especially the hospitalization time has been reduced significantly from months down to weeks. This is mainly due to the treatment called Baby Big. And Baby Big is and it's a drug that is based off of human-derived immunoglobulins from people like myself. I was able to contribute to this project, and we were immunized versus against botulinum neurotoxins. So they would collect our plasma, and they would be able to use our immunoglobulins to give us to a patient that has infant botulism. And so it was dramatically reduced the number of being able to treat numerous cases of infant botulism, but it's reduced the amount of money from hospital costs, et cetera. So, I mean, it's really been a great drug, and it reduced the a number of deaths down significantly. And this was all developed from the California Department of Public Health, their infant botulism treatment and prevention program. So they're the cutting edge researchers there. So now we're going to move into the current status of the by heart recall from powdered infant formula and the infant botulism cases that have derived from that. So way back in August of this year, from August to November 8th, the California Department of Public Health, they noticed an uptick of infant botulism cases, well beyond what is normal based on their surveillance. And 36, so they had detected 84 infant botulism cases that were under treatment. 36 of those had consumed powdered infant formula, and 13 of those cases consumed By Heart brand specific powdered infant formula. And By Heart contributes about 1% of the market share. So this was a large number of cases attributed to this kind of a product. So that's why By Heart then initiated a recall of 2 lots of their product. And then on November 11th of 2025 of this year, the California Department of Public Health, their Infant Botulism Treatment Prevention Program, IBTPP, they detected a preliminary positive type A toxin producing strain in an open can of powdered infant formula that was taken from a patient's home. Now, samples from patients' home are provided voluntarily. They are not seized anything from the patient's home, but they are offered up voluntarily for testing. And so they did detect type A producing strain, not the toxin, but the strain of the organism from the open can of powdered infant formula. So By Heart then expanded the recall of all of their products. And by November 15th, the number of cases increased from 13 to 15. These were all hospitalized patients. There were no deaths, thankfully. And the age range was about 2 weeks to 5 months, so 16 days to 157 days. By November 14th, the number of cases increased to 23 and then up to 31 by November 19th. And as part of the FDA update, because they have been testing products, finished product sampling, as well as I'm sure they're doing ingredients and environmental samples as well. But they put a statement out that says detection of Clostridium botulinum in infant formula is complex, and a negative test result does not rule out the presence of the bacteria in the product. So that's an important statement that we're going to show because we're going to talk about that in more detail in a subsequent slide, but I just wanted to highlight that. So just because we didn't detect it necessarily, it could still be there. So November 20th, recalled product was noticed still on the shelf after this outbreak and after the product was recalled. These are in prominent retailers like Walmart and Target and Kroger locations and things like that. So our lab, we were actually able to get some off of eBay. Several of the orders, though, were canceled, but we're able to obtain some product and help get some off the shelf from eBay. So by November 26th, the number of cases increased to 37, and this is across 17 states. So keep in mind, this is not just within the United States. It's expanded all throughout the world. This is a globally distributed product. Um, so then FDA also did release their FDA establishment inspection reports and their Form 483s, their inspection observations from 2022 through March of 2025. And these were mainly from the three blend house manufacturing facilities. So I have pictures of them on the right-hand side there from top to bottom, the Allerton plant in Iowa, the Portland, Oregon plant, and the Reading, Pennsylvania plant. I'm not going to go through the 483s or all of the details of all the findings. They're available for the public to read through. They're heavily redacted, but you get the gist of what was going on. So they noticed some GMP deficiencies. There was some potential exposure of the product to as being adulterated. By December 10th, the most recent update, and I just checked it this morning, it's still the December 10th update. The case definition was expanded to include any infant with infant botulism who has been exposed to ByHeart formula at any time since ByHeart released their products in March of 2022. So this identified immediately then 10 additional cases from December of 2023 through July of 2025, bringing that total number of cases to 51 cases, 51 hospitalizations, and through 19 states. So in this table below, this shows that the California Department of Public Health had identified a strain of Clostridium botulinum that produced type A toxin. And ByHeart also did some finished product testing of some of their batches, and they found there were 5 samples that tested positive for a type A botulinum toxin producing strain. And so this is, again, still an ongoing outbreak. So I don't want to speculate as to what is the root cause of this at this point. But we're going to go through some of the historical considerations. So has powdered infant formula ever been the cause of an infant botulism outbreak before? That's still kind of debated, I think, and to some of the researchers in the research community. But so I'm going to highlight a case that happened back in 2001. So in the United Kingdom, back in 1978 through 1994, they had 5 different reported infant bot cases that were identified without any kind of a source that was identified as what was attributing to those outbreaks or those 5 cases. In June 9th of 2001, there was an infant that was a formula-fed infant, a 5-month-old, that had infant botulism. And so when they went through the investigation, they had detected type B toxin producing strains from the rectal washout and the feces. So on the table, hopefully this is clear enough to see. This shows, what the number of enrichment cultures per food sample, but then the results of the enrichment. So the isolated type B strains from the enrichment cultures from the infant formula milk powder of unopened cans, but also in open containers, and also from the rectal washout and feces of the actual patient. So in the open container, they had identified some additional isolates, and they estimated that in the open container that the infant was being fed from, that amount of spores were about 0.38 spores per 100 grams. In some of the unopened containers that were tested of similar lot, they found separate C. botulinum type B isolates, and they estimated that content to be about 0.14 spores per 100 gram in that case. So when they looked at these strains and did a little bit more of a comparison of the strains of the isolates, because the idea in any outbreak situation is to match the patient isolate with the product isolate. And in this case, the open container, those type B isolates, they were analyzed through amplified fragment length polymorphism, and they found that the isolates were comprised of different banding patterns or different patterns, patterns 1, 2, 3, and 4. And the pattern 1, the strains that had the same pattern of pattern 1 were the same as the patient isolate. So the patient isolate also was of pattern 1, 1 and 3. So these isolates did match in the open container. The unopened container, however, the isolates from that container were of AFLP pattern type 5, which then did not match the patient sample. So the authors of that paper concluded, though, that it's possible for the presence of a mixture of C. botulinum isolates in a single product, And as the infant was consuming powdered infant formula, the certain strains may have colonized better than others. And so when they were able to detect the organism from the patient, they just detected the different AFLP pattern type. Another study that was conducted by Eric Johnson here at UW-Madison around that same time with those specific isolates, they did pulse field gel electrophoresis, and they concluded that the isolates from that outbreak the product isolate in the unopened can did not match that of the patient. And for sure that they concluded that no, in fact, that powdered infant formula was not involved in causing that outbreak or that case of infant botulism. So a survey was done shortly after that in 2009 by Jason Barish, Dr. Barish, over at the California Department of Public Health's IBTPP. They surveyed 30 powdered infant formula samples, and they revealed that although none of them tested positive for Clostridium botulinum, they did test positive for Clostridium sporogenes, and they found the spore counts to be ranging from 2.2 to 6.9 per 100 gram. So these were the 30 powdered infant formula samples that they assayed. These were from cases of infant botulism. So the infant did have infant botulism, but they didn't actually isolate Clostridium botulinum from the actual powdered infant formula, but they were able to look at spores in general, like what was there. So they detected Clostridium sporogenes. They also detected multiple other different species of Clostridia. So when they also looked at the spore load of sporogenes at 2.2 to 6.9 per 100 gram, they suggested that a typical patient would ingest about 2.2 to 7 C. sporogenes spores every 24 hours. And as I pointed out earlier, Clostridium sporogenes is a very close genetic relative to group 1 C. botulinum, so they likely share the same environment. And so it's possible that if sporogenes is there, it could also mean that Clostridium botulinum is there. And there are 21 strains of C. sporogenes that I pointed out earlier that have been found to produce a serotype B toxin. One other thing they did find is that they have a wide array of different Clostridia that was identified in the powdered infant formula screening. Different organisms like Tyrobutyricum, which has been attributed to late blowing of Swiss cheese, Butyricum strains, perfringens strains, which perfringens is another pathogen. So this the type and the amount of Clostridiospores varied depending on the brand. So they also included some samples that were pulled off of the shelf as like sort of a market surveillance to include and not just patient samples only. So it did vary with brand to brand. So there was some information that would suggest that the contamination level of milk, so if it was to come in on the milk, the contamination level is extremely low, like 1 spore per liter. That was some of the information, but there's not a lot of studies that have been done to show what the prevalence of C. botulinum spores are in milk. So that's really unknown, other than this one suggested sample that was saying 1 spore per liter. So around the infant botulism cases to look for what could be the cause, dry cereals, non-fat dry milk, canned formula, bottled baby food, commercial baby food, some of these things were submitted in a survey to kind of see what the prevalence of C. botulinum spores would be in those products. And out of all of those, only one out of those samples were tested positive for a C. botulinum type B strain. And so if you look back to that study from the 2001 UK infant bot case, so based on the level of spores that were found in the unopened cans in that particular investigation, the authors in that paper suggested that at least 200 grams would be necessary for a 95% chance of detecting a single C. botulinum spore. So based on the sample sizes that are presented here and the number of samples that are presented here, it's possible that in these earlier surveys conducted in the 1980s that we were just simply missing it by not including a high enough sampling amount, sample size and sample numbers. Another point I also wanted to bring up was the 2013 Fonterra recall. This was a big deal in the news. Testing by Fonterra, they were making a whey protein concentrate, and they detected the presence of Clostridium. in contaminated whey products, and so they immediately recalled the product. There were no cases of botulism that were actually reported, and the later testing actually revealed that these were non-toxigenic strains of Clostridium sporogenes. So this created a huge problem with regard to this company. I mean, at the economic cost of hundreds of millions of dollars in the recall, as well as litigation and settlements and things like that. So testing for the presence of Clostridium, they found Clostridium sporogenes. So is that something that we want to think about going forward, or what are some of the regulations that maybe should be changed or not changed, or what should we do in the future to prevent these kinds of things from happening? So based on the 2001 UK infant bot case, there were numerous risk assessments that were conducted by the World Health Organization, also by the UK Food Standards Agency. And the World Health Organization, based on some of these risk assessments, they came out and said basically causality is less plausible or not yet demonstrated. So meaning Clostridium botulinum, yeah, it may be detected or identified in a powdered infant formula, but it's not yet been implicated in causing illness in infants. And so these risk assessments, when you look at it, the value or the amount of spores that are present is like 0.3 spores per kilogram, up to 10 spores per kilogram. So 0.001 to 10 spores per kilogram. So relatively a very, very low risk of causing disease. And so they maintain that this is a low risk. There's no need to test for Clostridium botulinum, or it's not in finished product. We don't typically do that anyway for any kind of a food product. We just assume that it could be there. But based on this, we have billions of servings of infant formula that's delivered, to or consumed by infants. But we don't have billions of infant botulism cases. So the epidemiology kind of matches sort of the prevalence of spores. So in the summary, there have been an increase in infant botulism cases over the last decade, both in the US and Canada, and presumably other countries as well. Historically, there is a low prevalence of spores of botulinum producing Clostridium in powdered infant formula, as I've shown. But it could be that the powdered infant formula could serve as a potential source, but we need more studies conducted to actually identify Clostridium botulinum in powdered infant formula and what is actually the level by maybe using a higher volume or higher sample sizes instead of what was tested previously. I do want to caution that the outbreak investigation by BioHeart is still ongoing, and although determining a root cause is vital to prevention of future outbreaks, it would be very, I guess, immature for me to provide any kind of speculation as to what could be the source. Is it sanitation versus the ingredients? Is it coming in? Where are the botulinum spores coming from? I think we won't know that until the actual investigation is completed. And so we have great researchers at FDA, CDC, California Department of Public Health. They're all working together to find out the answers, and by heart also. Whole genome sequencing of the patient and product isolates kind of presents a unique challenge. Because there could be the potential for more than one isolate to be identified from the infant botulism cases over the course of 3 years. Depending on what is the source of these outbreaks, it's possible it's not going to be just one isolate, so the isolate in the product matching the isolate in the patient. So similar to that UK outbreak case, we could be dealing with multiple different strains. There was a case too, and so even though type A was identified, there could be additional types of toxins that could be identified in some of these additional patients that come about that could be producing type B toxin. In 1975 and through 1987, there was a screen of different isolates from fecal samples that was conducted by CDC of 336 infants, and they found one case, I think it was a Hawaii case, where the infant actually had two different types of organisms isolated from the fecal samples. And that was a type A producing strain and a type B producing strain. So it gets to be more complicated than just having a single strain that's going to be identified from this outbreak. So lastly, just to kind of give an idea of what my lab does, I run the Applied Food Safety Laboratory. This is a lab that was run by Kathy Glass, if you know of Kathy Glass. But it's at the Food Research Institute at UW-Madison. We conduct food challenge studies. We work with the food industry to help validate the safety of their food formulations. We're more formulation process authorities than thermal processing process authorities. But we also work with regulatory agencies like FDA. I used to work for FDA. So I love my friends there at FDA. And so we work with regulatory agencies to either change or support certain food regulations. And again, we work with the pathogens that are listed here, but namely Clostridium botulinum, which our lab is a tier 1 select agent laboratory. There aren't many labs that work with the actual organism and the toxin. But we're fortunate we have two different labs. So our lab does all of the applied microbiology, and then the Dr. Pellet lab, which used to be the Dr. Eric Johnson lab, they do all of the molecular work and whole genome sequencing of botulinum strains. So that is me in a nutshell, and that is my presentation. I'm a little bit over time, but our next speaker is going to be Dr. Snyder, and she's going to be talking about sources, potential sources of spores, right?
In part, you may be overselling what we can really conclude at this point with
Right.
that. Yes. So I'm going to talk about cross-contamination. I'm also going to talk about consumer risk messaging and some ongoing work there. So, in instances of product contamination, generally speaking, contamination could occur from a few sources. So, it could, number one, come in on raw materials. It could be introduced in the production facility through cross-contamination, or three, it could be introduced um, after the product is opened by the consumer through cross-contamination. And so, I'm just going to spend a couple minutes talking through those possibilities, and we'll do it first for Chronobacter. So Chronobacter is a vegetative cell. It doesn't form a spore, so it's going to be sensitive to thermal processing, meaning Chronobacter will be inactivated by pasteurization. So although it's possible Chronobacter comes in on ingredients, if the ingredients are blended and then pasteurized before spray drying, that thermal process will eliminate Chronobacter from those materials. The exception would be if there's ingredients blended in after the pasteurization step. So for a lot of powdered infant formula, it is post-pasteurization recontamination that's the concern. And so this can happen in a production facility at any step downstream of that pasteurization treatment if there's not going to be a subsequent kill step. And so to address this, formula manufacturers conduct environmental monitoring to eliminate Chronobacter species from their environment, and they also test finished product for Chronobacter as well as Salmonella per 21 CFR 1, Part 106. Now, an exception to this case where post-pasteurization recontamination could occur would be in product that's sterile, so ready to feed liquid infant formula. So that product is shelf stable and the risk of environmental cross-contamination in the facility is exceedingly low because of when and how the kill step is applied. It's considered commercially sterile. Um, and then finally, Chronobacter contamination of PIF can occur after the product has been opened by the consumer. So field sampling studies can find Chronobacter in households, and there have been a couple case reports where cases of invasive Chronobacter infections in infants have been linked to contaminated formula or expressed breast milk, suggesting that the cross-contamination, though, occurred at the household level specifically. So identifying the differences there in sporadic cases, meaning one-off cases between household contamination, post-pasteurization, recontamination in the production facility is difficult. So in those one-off cases, there's often more limited evidence to define where contamination has occurred. So sporadic cases being different though than instances where you have systemic contamination, where a number of product units are contaminated over time. So you have multiple people sick and there's an epidemiological signal. So comparing this with Clostridium botulinum, big difference here, CBOT's a spore former. So CBOT spores are not sensitive to processes like pasteurization. So the spores on incoming materials are not going to be eliminated by pasteurization. Could it be introduced through the production facility via cross-contamination? Perhaps. There's little prior evidence of this for many components of this case. But to get growth in the environment, you would have to have the minimum conditions for CBOT growth as you would in any other circumstance. So it's an obligate anaerobe for one, and you would need sufficient water activity and temperature to get growth under those circumstances. And then finally, could you get CBOT from consumers' home via cross-contamination? Yes, though not likely the explanation in this case. I say yes because there are prior cases of infant botulisms that have been attributed to dust or soil-borne spores in the infant's environment. But I say not in this case because there is an epidemiological connection, right? The recall updates have described the number of cases of infant botulism where the baby was fed by heart compared to the percent of formula market share by heart in the US. Also, more recently, ByHeart reported the finished product testing contamination in an open can that Kristin discussed. To be clear, as we've said, you need the results of the investigation. And just to state the obvious, there's still information we need from that investigation before we can define root cause here. Looking into the ingredients in ByHeart's formula, you'll notice there's a number of major and minor components. Major ingredients include dairy ingredients, minor ingredients include oils. There's a number of vegetable oils listed here, sunflower and rapeseed and coconut and sunflower, vitamins and minerals. Some formulas will include starches or vitamins, for example, can come in carriers. You'll also note that this formula was marked as organic, and a number of the ingredients here are identified that way on the product label. So I showed this label not to be alarmist about like a long list of ingredients. Formula is a sole nutrition source for infants, so it's going to have a list of ingredients. But I'm just illustrating what ingredients are common in formula and what specifically was in this formula. Spores can be found in a number of environments. I mentioned homes before, so there are, like I said, reports of CBOT spores linked to infant botulum cases from home environments. But overall, there's relatively few sampling surveys that have been done for CBOT. So many of the surveys that have been done are conducted in natural environments, so from soil or sediments or dust, some from agricultural environments, and some from aquatic environments, particularly for toxin type E, Clostridium botulinum. From foods, there have been surveys across honey, I think most notably for its previous association with infant botulism cases, some dry plant materials like spices or grains or teas, and then there's some data on infant foods, which Kristin largely summarized. In infant formula, like she said, prior evidence of detection of CBOT spores was rare, and if present, it was at low levels. So that evidence, coupled with the fact that prior to the ByHeart case, there's been an absence of this sort of epi signal. So specifically that this is perhaps the first outbreak linked to PIF. So as Kristin described, a number of prior assessments concluded that the risk of infant botulism linked to formula was low. So as the investigation evolves, it will be interesting to identify what has changed here in this case. For minor ingredients like the starches or the oils, there's definitely a lack of field sampling surveys to give an indication of prevalence of CBOT spores in those ingredients, but those ingredients are also highly refined. So again, the thinking has been that the potential risk from those ingredients was low. I'm going to talk briefly about some of the consumer food safety aspects. As you know, a number of consumer groups recently wrote a letter to federal agencies calling for a response in this case. They asked for restoration of funding and staffing for federal agencies, ensuring effective recalls. Again, as Kristin discussed, the FDA continues to report sort of isolated cases of products still being found on shelves, and enhance regulation around CBOT and powdered infant formula. Relatedly, on Monday of this week, FDA released an open letter about the responsibilities that attach for effective recalls, and FDA sent warning letters to 4 retailers for failing to remove recalled ByHeart product from shelves. And so in this letter, it says that regulators checked retail stores across the US over 4,000 times to ensure that the recalled product was not available, but they found recalled product in store shelves over 175 locations across 36 states. I want to compare a little bit about some of the differences in risk messaging for consumers. So Chronobacter and CBOT, in both cases, the goal of that risk messaging is to get consumers to take action regarding discard of the recalled product. Also, in both cases, the goal is to not panic consumers into responding by adopting less safe practices, like preparing their own formulas at home. There are reports of this happening. I have a screen grab here from a website that recommends this. For Chronobacter risk messaging, part of the goal is helping consumers recognize the definitions for babies that are at high risk for illness. On the CDC infographic, you can see it defines risk groups here based as infants in the first 2 months of life, immune compromised, or born prematurely. So caregivers for that group are the ones who may need to adopt extra precautions to protect against Chronobacter illness. And in this case, extra precautions could include hot water reconstitution of this or the use of sterile formula. And so this is in addition to general good hygiene practices like hand washing and surface sanitation to prevent household cross-contamination. By comparison to the messaging regarding infant botulism, there are some similarities. So still we want people to take action in discarding recalled product. We don't want them to make their own formula. We do want them to use hygienic practices. And so that makes sense. But for infant botulism, that risk group is different. So now babies up to 1 year of age are considered at risk for infant botulism. Of the cases implicated in the current outbreak, I think the oldest is around 8 months of age. But and so you may be aware, right, that recommendation for honey actually is for infants 1 year of age or less. But a notable difference here is that the hot water reconstitution is not going to help because Clostridium botulinum is a spore. So fewer interventions at the consumer level, but sterile formula remains an option. Just a bit about those recommendations. You can see an example here of products. The bottom here is this ready-to-feed liquid formula. It comes in small bottles and it has a processing control in place for Chronobacter and Salmonella and Clostridium botulinum. An additional advantage is that it doesn't require mixing with water. It's ready to feed. So this is unrelated to microbial food safety, but it's solved by ready to feed formula. Caregivers don't have to reconstitute the powder with water. So I suppose there's some advantage there for microbial food safety. There's less opportunities for environmental cross-contamination, but it also reduces excessive dilution of the product, which can happen because formula is so expensive. So from a food technologist perspective, this is a great choice. It solves a lot of problems. The trade-off here is that it's expensive. So a blanket recommendation for consumers to use sterile formula, even for certain groups like those sensitive for Chronobacter, is difficult because that cost trade-off is significant. I'm showing here just some estimations for cost per ounce of the reconstituted formula for different products. These are sort of one-offs and they're not the only or perhaps even the average cost basis, but just to give you some sense of differences here. So comparisons between a readily available brand for the powdered infant formula, 54 cents per ounce compared to the ready-to-feed formula at 83% cents per ounce. And this might seem like a modest increase if you break it down per ounce like this, but multiply it across the cost of what a family would need to pay per month for an infant who is 100% fed by formula. And that's an expensive monthly bill. Compare it though, however, to specialty brands like By Heart. It's not as expensive as that is per ounce of reconstituted product. So it may be accessible for some consumer groups. The cost increase is certainly important, but it can be an option. And so one that I think caregivers should be aware of that it exists. And so I'm going to conclude by sharing my lab here has a special focus on microbial safety of infant foods and feeding practices, in particular for formula and human breast milk. And we are going to take questions at the end. You can feel free to reach out to any of us individually for follow-up. But with that, I'm going to pass it over to Nicole.
Thanks, Abby. And I'm going to finish up pretty quickly here with a short overview of the types of microbial testing that are commonly done in infant formulas, in powdered infant formulas. So I'm Nicole Martin. As Abby said earlier, I'm an assistant research professor in dairy foods microbiology. My area of expertise is in dairy food safety and quality from a microbial perspective. And I have a particular interest in spore forming bacteria, so I spent a lot of time working on that over the last 20 years. So first of all, let's talk about pathogens or potential pathogens that are tested for. Obviously, the two biggest ones here are Chronobacter and Salmonella. These have resulted in numerous outbreaks, as both Abby and Kristin have already discussed at length today. And then we have a number of other pathogens that are tested for that we would consider low to medium risk pathogens because they are relevant to infants but not previously identified in infant formula or not having caused an outbreak. And those include Listeria monocytogenes, E. coli, coagulase positive Staph aureus, and Bacillus cereus. You'll notice that Clostridium botulinum is not on here, and that's for a number of reasons that have already been discussed, but also because it is challenging to test for specifically. And then there's a broad range of indicator organisms, and this varies a lot over different sort of regulations for different countries. But generally they're going to include a specific set of organisms. But let's just remember the definition of indicator organisms here. These are markers that reflect the general microbiological condition of a food or environment. So these are not specifically associated with the presence of pathogens. They tell us more about was there a lapse in GMPs? Was there a hygiene issue in this food? Were there conditions that allowed for the growth of specific groups of microorganisms that would tell us about the process that this food was manufactured under? So again, this is a pretty lengthy list that includes aerobic, mesophilic, and thermophilic plate counts. So these are sort of a general hygiene indicator. Coliforms, fecal coliforms, and Enterobacteriaceae. Obviously, these are important groups for a number of reasons, but in particular, EB or Enterobacteriaceae, because those pathogens that we're really concerned about fall within this taxonomic group. And then a couple of different types of spore formers, and honestly, I've seen these, even more different types of spore formers tested here, but mesophilic and thermophilic spores. And then the last one here, which is sulfite-reducing Clostridia or SRC. This, I just saw a news article that a formula manufacturer started testing for SRC, and they wanted to get the news out there because this is something that can be done as an indicator for Clostridia presence in formula. Some manufacturers do SRC testing and some don't. I don't think this is probably something that lots of folks are doing, and there's a couple of reasons for it, and I just wanted to talk a little bit more about SRC. So just generally, though, the process for testing for sulfite-reducing Clostridia is to first heat treat the sample because we're looking just for spore formers here. So we want to eliminate any vegetative sulfite-reducing bacteria, which there are many. So heat treatment to eliminate those vegetative cells, and then plating on an iron sulfite agar. And then incubation under anaerobic conditions. This, again, we're selecting for Clostridia, or what we want is Clostridia here. And then enumeration of typical black, gray, brownish colonies, which would be an indication of the reduction of sulfite to sulfide, and then reaction of that sulfide with the iron compound in the media. And then, technically there should be a confirmation of these colonies for a strict anaerobiosis, which would indicate Clostridia. And I'm going to tell you why there should be confirmation. I don't think that is typically done, however. But it's because there are a number of spore-forming bacteria that are commonly found in food products that can both grow under anaerobic conditions and reduce sulfite. And this study out of Paul Cotter's lab at Chuggas did some characterization of sulfite-reducing bacteria in dairy products. And of the 101 isolates that they selected, about 25% were not Clostridia. So that's why that additional confirmation step is really important, because this is a fairly broad group of organisms that can produce typical colonies on this iron sulfite agar. So this was a small study, a fairly small study, but they did about 25% of the isolates were not Clostridia. And of the Clostridium that they isolated, none were botulinum. So there's a wide variety of Clostridia that are able to also produce colonies on this media. But again, this is an indicator test, so this, may be a said indication that there are conditions that would allow for the growth of sulfite-reducing Clostridia in food products. So that's it. If you scan this QR code, you'll get my contact information if you want to reach out with questions or with testing inquiries. My lab is predominantly focused on dairy microbiology, and we do some industry testing for groups who are interested in that as well. So you can go ahead and get my info from that QR code, which, maybe that's weird at this point, but I thought it was convenient. So, all right, I think we have a few minutes for questions. Um, Abby, I'm going to turn it back over to you.
Yeah, we do. You can also Google Nicole to find her. You don't have to use the QR code, I suppose, if you don't want. We'll make sure if you have follow-up questions, you could reach out to any of us individually. So we're probably not going to get through all the questions that we had pre-submitted, which is fine, but we'll get through a few.